And first up, we have Natasha Ufer from University of Queensland, who's going to be talking to us about establishing framework species in bauxite mine rehabilitation in Northern Australia.
My name is Natasha, and I am presenting some of our work from the Ufer region, where we were establishing framework species in bauxite mine rehabilitation. I acknowledge the traditional owners and their custodianship of the lands in which we meet, and I pay my respect to their ancestors and their descendants. This map is showing the Ufer region, where bauxite has been mined by Rio Tinto since the early 1960s. And 30,000 hectares of vegetation has been cleared in this time, with another 27,000 hectares set to be cleared in this area south of the Embley River down towards Arakoon. Our study site in this region was the Hay Point bauxite mine just across the river from Nappanum, and it has been mined by Green Coast Resources since 2017. This work could happen under the Advanced Queensland Innovation Partnerships project for Indigenous employment, forestry livelihoods and mining. And this was conducted by USC in partnership with UQ. The final land use goal on this mine was a self-sustaining native plant community resembling unmined neighbouring forests with potential to support Indigenous livelihoods by providing future opportunities for harvesting of timber and non-timber products. After consulting with the traditional owners, we decided that these were the framework species for the rehab to focus on. And by framework species, I mean those that form the main structure of a vegetation community. So we had Eucalyptus tetradonta, Corymbia necrophylla, and Erythrophilium chlorastaceus, the three framework tree species of the region, as well as Acacia rothii and Parinari nonda, which is the nonda plum, and a coral tree, Adenanthera pavonina, that was not a framework species on the mine, but we included it for its cultural significance. The seeds of these species were collected by ourselves and traditional owners on the mine and in the surrounding Ufer region. This is showing the mining pit that we rehabilitated, where soil was stripped before mining and stockpiled in 2017 and 2018. This mixed grill topsoil that you see in the middle is a mixture of both of these other soils as well as the underlying subsoils. And this is pretty common practice for soil handling in the region. And this map on the right is showing these topsoils after they were respread for the rehabilitation. We split this rehab pit into two trials. The top larger section was for tube stock planting, and we only included the three framework species in this trial as it was for future timber harvesting. And we also planted trials with superphosphate fertiliser and biochar that we sourced from the southwestern Australian Jarrah eucalypt forests, where bauxite mining also occurs. And this southern section we used for a direct seeding trial. And direct seeding is the standard practice for the Ufer region. And we also use the region's standard superphosphate fertilising rate of 200 kilos per hectare. And we also considered the two topsoil types in this area, which were the 2018 topsoil and the mixed grill. Before rehab, we needed to send some of the seeds of the three framework tree species to the Gulkula mine in the Northern Territory. Gulkula is an Indigenous owned and operated mine, and they grew these seedlings in their nursery for 4 to 5 months before they were transported by plane back to Ufer and by boat across the river to the Hay Point mine. We planted these in the tube stock rehab in February last year, planting the two framework eucalypts in rows with the fertiliser that you can see in yellow, and with the combination of fertiliser and biochar, then the biochar alone, and the green is showing our control treatment where we didn't plant with anything. On site, however, there was this pretty large waterlogged area that we couldn't plant in. So we instead decided to plant all of the ironwood seedlings around this area due to them being a little more water tolerant than the eucalypts. So they weren't part of our trial with the superphosphate and the biochar. But in this soil treatment trial, we planted the seedlings with a standard amount of the soil treatment required. So for biochar, that was about 65 grams, and for superphosphate, that was about 30 grams. We collected data here in September last year at about 7 months post-planting. When we arrived on site, we saw that fire had burnt a lot of the tube stock area. But we still needed to collect data, so at each tube stock plant, we recorded if they were dead or alive. We recorded their height and gave them a health score between 1 and 5. And we recorded if they were resprouting after the fire. Some actually were, as you can see here, that's a bloodwood resprouting. Our results showed us that fire affected half of the planting area and burnt 1/3 of the seedlings. The ironwood, however, as we planted them all around the waterlogged area, they were completely unburnt by the fire as grass didn't establish well there and didn't spread the fire there. 15% of the fire affected Corymbia necrophylla and 12% of the fire affected Eucalyptus tetradonta were resprouting. That's showing another bloodwood resprouting here. And with satellite imagery, we could see when these fires occurred. And on this side, where the fire occurred 5 weeks prior to data collection, there were 82 seedlings resprouting. And on the other side, where the more recent fire occurred 2 weeks prior, there were only 2 seedlings resprouting. So we can tell that the seedlings do resprout, but it would usually take more than 2 weeks, and that when we left the site, more could have resprouted as well. Our results also showed that between all of the treatments and the other seedlings we planted without any soil treatment, the Corymbia necrophylla had the tallest seedlings on average, followed by the tetradonta, about 9 centimetres shorter, and the shortest seedlings were the ironwood at about 16 centimetres. When considering the soil trials, the soil treatment trials, Corymbia necrophylla had taller and healthier seedlings in the biochar and the combination treatments, and had shorter seedlings with lower health scores in the fertiliser treatment. Interestingly, the Eucalyptus tetradonta almost showed the opposite effect, where it had shorter seedlings with lower health scores in the biochar treatment and showed improved health and height in the fertiliser treatment. The direct seeding trial we sowed by hand in November 2019, and we sowed all of the species I mentioned before, also including the coral tree. And to broadcast this evenly by hand, we split the pit up into 5 metre wide sowing strips there were 40 of these going across the pit. And within each of these sowing strips, we wanted to make it even easier by dividing them into 10 metre blocks. So as we were meandering through, we knew how much we needed to sow in each of these blocks. We used a standard amount to sow in each block with kitchen measuring spoons, and we split up all of the seed into a small seed mix with the 2 framework eucalypts and a large seed mix using the remaining seed. However, the Nonda plum was too large to fit into these spoons, so we needed to sow it as about 1 to 2 Nonda seeds per sowing block. This is what the direct seeding trial looked like, and I studied this for my honours year. We applied the standard fertilising rate in 40 by 40 metre blocks at 2 months post-seeding. And we also established 37 assessment plots in each of these treatments with or without fertiliser, and in the 2018 topsoil region and the mixed grill soil region. We collected data at 2 months post-seeding at the same time as applying the fertiliser and at 9 months post-seeding. And in each plot, we collected the stem counts and the heights of the framework species, and also sampled soil from the top 10 centimetres, testing for these parameters. These photos are showing the same plot from 2 months post-seeding to 9 months post-seeding from the wet to the dry season. Our direct seeding results showed that between the soil types, so between the 2018 and the mixed grill, there was no significant difference in soil parameters, and also no significant effect on the establishment, that is the density and the heights of the framework seedlings. The fertiliser did affect the soil parameters, as it significantly increased electrical conductivity and significantly decreased total nitrogen. However, this still didn't show a significant effect on the framework species' early establishment. And this could be suggesting that the standard fertilising rate is too high, or at least not optimal for these species at this early stage of rehab. Our results also showed that the bloodwood Corymbia necrophila showed almost twice the growth of the other dominant species, Eucalyptus tetraodonta, and that the coral tree Adenantha pavinina was not likely to survive, as all of those we encountered were really dried out and didn't look like they were going to survive. In conclusion, this study has shown that tube stock is suitable for establishing framework species in early rehab, but it could be more viable to use where seeding isn't working very well. Our tube stock trial showed that biochar and fertiliser can improve tube stock growth, but it could be dependent on the framework species planted. And our direct seeding trial showed that fertiliser did not actually improve the direct seeding establishment in this early stage, and that it could depend more on the field performance of each species or their regeneration potential from the topsoil seed bank, which could even change from site to site. We found that the coral tree is probably not suitable for early rehab, but that's not to say that it couldn't be established at a later stage, perhaps even by tube stock. And both of our trials are suggesting that Corymbia necrophila could be an easier framework species to establish than Eucalyptus tetraodonta, the dominant tree species of Northern Australia. The outcomes suggest that changes are needed to these current rehab practices at Weipa, and that more research is certainly required, especially around species establishment requirements and how the best practice soil handling and different fertilising methods affect the early establishment of framework species. Thank you for your time. I want to acknowledge those who were involved in this project, and they were John, Mark, Peter and Gabriel, the Advanced Queensland project with USC and UQ, the WIC traditional owners and Indigenous community members for their help on site and for seed collection, Robin for her field assistance throughout this project, as well as Taylor and Norma for their assistance with data collection. Thank you for listening.
Working? There you go. Thanks for that, Natasha. Very interesting project and with some intriguing results. Is Natasha online to answer some questions?
Yeah, I'm here. If I can be heard, I can answer questions.
If Natasha is there, I've got a question. But, oh, yeah, there you are. Hey, Natasha. Does anyone else on the floor have a question before I go? Yep, sure.
Thanks, Natasha. Just a curious question about what caused those fires? What was the fuel? Why did those areas burn that caused some damage? And what would be the likely need for future controls, maybe a fuel?
As far as I know, I think one might have been an escaped control burn, and I'm not sure about the one on the other side, but yeah, I think if the grass had been controlled in that rehab pit, it probably wouldn't have spread through as much of it.
Ping Lu from EI. Just a comment, actually, and Natasha, it's very good work. And usually the Eucalyptus tetradonta are slow-growing species in the first 4 or 5 years. So you might expect that it's normal to have that growth rate. And they will pick up after 5 years, because comparing, for example, I don't know whether you have miniata at your place, and compared to miniata, the tetradonta is much slow growing. So the miniata will be fast growing in the first 5 years and slow down. And the tetradonta will be, in contrast, will be slow in the first 5 years and then pick up. That's just comments for you.
Yeah, that's really interesting. Thank you. Yeah, because this was just a really early assessment, only 7 or 9 months after the planting and seeding, we would really like to go back at, 5 years would be good, and we could see what's happening then.
More questions? Yeah, Chris?
Thanks. Given you were looking at potential for timber, did you look at whether trees were growing straight and were going to be suitable to be timber trees?
Yeah, we did plant them with those sticks, and I don't really know what they're called, but the clips to keep them straight. But yeah, we didn't really do any assessments for how well they were going to grow into timber trees, although that was the idea of planting those there.
Any other questions? No. Got time for one from me. Just going back to the sort of what Ping said, and tetradonta growing slower than that Corymbia nesophila, putting your plant physiologist hat on, do you have any idea of why that might be? Like, have any guesses? Obviously you're you know these species better than most, so.
I don't actually know. We have done some glasshouse experiments when we did the seed viability testing, and yeah, I don't actually know much about how well they grow after the early stage now.
Yeah, thanks. That was the main question. Well, Ping's got. Oh yeah. Maybe we'll move on. I'll have a chat with you after. Curious plant physiologist. Thanks, Natasha. That was great.
Thank you, everyone.